A dull grey blob, the size of a pinhead, floats in a petri dish on a lab bench. The blob is made up of thousands of living, communicating brain cells. This is a cerebral organoid, and neuroscientists across the world are growing these structures in the lab to better understand the human brain.
The way that we accomplish this is by starting with stem cells, and then we essentially provide the right sort of environment to support growth of brain tissue, but not other organ types. So that what ends up happening is you have these 3D, balls of tissue that look a lot like the developing human brain in certain ways.
This is Madeleine Lancaster, an organoid researcher from the University of Cambridge in the UK. Madeleine was the first person to grow a brain organoid back in 2010, and now she uses them to study brain development and disease.
The human brain is hugely enlarged and very complex, and that gives us our unique cognitive capability, but it also makes us more susceptible to neurological conditions that are not very well modeled in, for example, mice. So brain organoids are especially useful for looking at human-specific features of the brain and for modeling those types of neurological conditions.
Right now, researchers can't grow brain organoids more than a centimetre in length, and they lack much of the complex internal structure of fully formed brains, even those of simpler creatures like fruit flies. But as the field matures, some researchers are working on ways to increase these organoids' size and complexity, which raises an important question. Could such bundles of brain tissue ever become complex enough to be conscious?
I don't think it can be ruled out that a cerebral organoid could achieve consciousness. I think it's entirely possible that as organoids develop in complexity and similarity to human brains, that they could have conscious experiences.
Anil Seth works on cognitive and computational neuroscience at the University of Sussex in the UK. Anil thinks that if organoids were to become sufficiently complex, the activity they display could become more similar to that of conscious humans. The problem, according to Anil, is that we don't know exactly what kind of complexity could lead to consciousness.
You could say that being more similar in terms of neuroanatomy and neurophysiology to human brains, well, that's the right kind of complexity. But then there's also the possibility that you can have organoids of some kind of complex structure that's different from the human brain, but that is also sufficient for consciousness.
And this raises an even bigger question. If a future organoid were to develop consciousness, how could you tell?
The real challenge with organoids, of course, is they're just isolated mini brains. They don't have any natural behavior. So the only way to make an inference about whether an organoid might be conscious is through the analysis of its brain dynamics. We can measure the electrical activity of a cerebral organoid and compare it to the kinds of patterns that we see in different conscious states in humans.
Assessing consciousness in humans is tough. Alongside measuring electrical activity, brain imaging of patients in vegetative states has also revealed conscious-like brain activity. But neither method is completely foolproof. And even if there were a clear marker for consciousness in humans, that same marker might not indicate consciousness in organoids. That said, were organoids to exhibit the kind of activity seen in the brains of conscious humans, Anil would be concerned. Although an organoid's experience of consciousness might be very different to our own, they might still be able to experience some form of suffering and be unable to communicate it. That possibility leads Anil to believe that the field should consider guidelines for this kind of research.
Because organoids are rapidly developing, and not only in how sophisticated the technology is, but in how many labs are conducting this kind of research. And because we don't know any definitive way of assessing the conscious status of organoids, there is an imperative for ethical frameworks to be developed preemptively.
Anil is by no means the only researcher calling for guidelines in the field. Already, a committee of the National Academies of Sciences, Engineering and Medicine in the US is examining the ethical issues associated with organoid technology. They will publish their conclusions on any possible oversight mechanisms in 2021. But Jean-Tine Lushoff, a philosopher and neuroethicist from Harvard University, thinks it would be premature to regulate the current field based on concerns around possible organoid consciousness.
I do not believe, even remotely, that cerebral organoids has anything that would even approximate what we call consciousness. There are regulatory questions, and those are the old ones, the classical ones, like where do you get the tissue from, the cells with which you start the process? Do you need to tell people for what experiments you're going to use it, and so on? But it is not in any sense specific for the question of consciousness.
Any meaningful regulation of the field, she says, would have to rely on a firm definition of organoid consciousness, which we currently don't have. Jean-Tine also points out that the very appearance of organoids, the fact that they look like mini brains, can trigger our moral intuitions where perhaps they're unwarranted.
When we see something that looks very much like a human brain in this case, we start to ascribe properties of a human brain and our ethical considerations about human brains. So I think that is important to consider and to bring us back to the reality of research when we talk about ethical appraisal of such experiments.
There are plenty of opinions as to how this field should be regulated. Back in the lab, Madeline welcomes conversations about where ethical boundaries might be drawn in the future, so as to safeguard against organoids achieving consciousness. But she's cautious about imposing regulations on the field too soon.
I think it's also very important to remember, we're having these discussions about organoids being potentially conscious or not, but there are millions of actually conscious human beings out there who are actually suffering from neurological conditions that have no treatment because we don't understand them, because the animal models have not given us the answers we need.
What's clear is that cerebral organoids allow researchers like Madeline to study human brain cells in ways they never could before. And while future organoids could become concerningly complex, there's plenty still to learn from the comparatively simple ones we have today.